What if I told you that the choices you make today could shape not only your health, but also the health of future generations? Welcome to the fascinating world of epigenetics, the study of how chemical modifications can turn genes on or off without changing the underlying DNA. In this video, we'll delve into how factors like diet, stress, and environment can influence our genetic expression, using Prader-Willi Syndrome as a compelling example of these powerful interactions. Prader-Willi Syndrome is caused by missing or inactive genes on chromosome 15. These genetic changes don't just affect one area, they disrupt key functions in the body, making this condition stand out in our genetic research. Children with Prader-Willi Syndrome face constant hunger, delayed development, and muscle weakness. The underlying cause is genomic imprinting, a process that controls how certain genes are expressed based on which parent they come from. This unique mechanism explains the condition's wide-ranging impact. Genomic imprinting is an epigenetic process that determines the expression of specific genes based on their parental origin. On chromosome 15, the maternal allele is typically silenced through DNA methylation, while the paternal allele remains active. In Prader-Willi syndrome, the paternal allele is either deleted or left inactive due to epigenetic errors or mutations. This loss of paternal gene expression interferes with crucial physiological processes such as hormone regulation and metabolic control, leading to symptoms like abnormal temperature regulation, growth deficiencies, and impaired energy balance. Because the genetic defect in Prader-Willi syndrome is often epigenetic rather than structural, it can go undetected through traditional genetic testing methods. This is where epigenetic testing becomes essential. By analyzing DNA methylation patterns on chromosome 15, we can detect whether the paternal genes, which should be active, are improperly silenced. Advanced techniques like fluorescence in situ hybridization and array comparative genomic hybridization allow us to visualize these abnormalities and confirm whether the paternal gene is absent or inactive. These tools provide a clear diagnostic pathway, offering precision in identifying the underlying genetic disruptions. Once the genetic error has been identified, we can better understand its impact on the body. One of the most critical regions affected by Prader-Willi syndrome is the SNORD116 gene cluster. This cluster plays a vital role in regulating hunger signals and muscle development. When SNORD116 is not expressed, the brain's normal response to hunger is disrupted, leading to hyperphagia, the insatiable hunger that defines Prader-Willi syndrome. Additionally, the lack of SNORD116 impairs muscle tone and growth, resulting in developmental delays and muscle weakness. These biological consequences show how a single epigenetic error can disrupt multiple systems in the body. Current research is focused on finding ways to reverse or mitigate the genetic disruptions caused by Prader-Willi syndrome. Scientists are currently exploring CRISPR-based technologies to reactivate silenced genes, particularly targeting the SNORD116 cluster. Another promising approach is the development of epigenetic therapies that aim to modify DNA methylation patterns, thereby restoring normal gene function. Though this is still in experimental phases, these advances can offer hope that genetic editing could one day correct the underlying cause of Prader-Willi syndrome, significantly improving the quality of life for those that are affected. One of the most exciting discoveries in Prader-Willi syndrome research is the potential for epigenetic errors to be reversible. Unlike permanent mutations in the DNA sequence, epigenetic modifications, such as abnormal DNA methylation or faulty imprinting, this can be potentially corrected. In Prader-Willi syndrome, where the paternal genes on chromosome 15 are silenced through improper methylation, there's growing evidence that reactivating these genes is possible. Scientists are exploring new techniques that target the methylation marks themselves, aiming to erase or modify them to restore normal gene expression. Epigenetic therapies are being developed that specifically target this reversible silencing. For example, drugs that inhibit DNA methyltransferases, which are enzymes that add methyl groups to DNA, are being investigated as a way to reactivate silenced genes. Moreover, CRISPR-based epigenetic editing is emerging as a tool to directly modify these epigenetic marks at precise locations in the genome, offering a more targeted approach. By restoring proper gene function at the epigenetic level, these therapies hold the potential to correct not only the symptoms, but the root cause of Prader-Willi syndrome, leading to more effective long-term treatments. Prader-Willi syndrome has provided many insights into how critical gene expression is for fundamental bodily functions. By studying the effects of errors in genomic imprinting, we are uncovering new ways to approach not only this condition but a range of genetic disorders. As research progresses, gene therapies hold the potential to provide long-term solutions for managing and possibly reversing the effects of Prader-Willi syndrome, offering hope for improved outcomes in the future.